Good evening and welcome to the Marion Miner Cook Athenaeum. My name is Sarah Sandbar and I'm one of the fellows here. From a human perspective, industrialization is a success story that paints a picture of leaps and bounds in development, technology, health, and knowledge. Yet from the perspective of Mother Nature, industrialization has brought with it the destruction of her forests, contamination of her waters, and pollution of her air. Chemical contaminants, most of which are a result of humans, can have serious impacts on health. Joining us tonight is Tyrone B. Hayes, a professor of integrative biology at UC Berkeley, who will examine the impact of endocrine-disrupting environmental contaminants on environmental and public health. In addition to teaching, Professor Hayes has worked at the National Institute of Child Health and Human Development, the National Institutes of Health, and the Cancer Research Laboratories at UC Berkeley. For the past 15 years, much of Professor Hayes' research has focused on the role of endocrine-disrupting contaminants, particularly pesticides, on environmental health, public health, environmental justice, and global amphibian declines. As always, I must remind you that audio and visual recording is strictly prohibited. Please join me in welcoming Tyrone B. Hayes.
Thank you. And I'm pretty loud. It's okay if I don't use I don't like to stand behind things. I'm going to tell you a story tonight. And before I do that, however, I want to start with this phrase. This is a phrase we were just talking at the table about many places, but I think it's one of my favorites. Zimbabwe's been one of my favorites. And this is a phrase that I learned while working in Zimbabwe. It's a Xhosa proverb, but I actually learned it from the Shona people. And loosely translated, it means people are people through other people. So I never give a talk. So I never present myself or my work. Without first acknowledging and presenting the people who made me who I am. But now something's happened. So now you can hear me, but you can't see. Uh-oh. Let's see what's happened here. Don't know how to. First and foremost, I always start with an acknowledgement and thank you to it was working. My family. This is an old photo. You can probably guess why they did not allow me to be in them anymore. But especially to my parents, my father, Romeo, my mother, Susie Hayes, for their love and support over these first 49, almost 50 years. And as a biologist, I wouldn't be here without them. To my wife, Catherine Kim, for her love and support. And these two, my favorite son and my favorite daughter, would not be here without her. And I showed this picture. This is also an old picture. I always show this one because it's a prom picture, which might be fitting because this shawl that I bought is called a ladies' evening prom shawl. But I don't know which I'm more proud of, that my son borrowed my tie for his prom or that my daughter borrowed my earrings for her prom. It was an equally proud dad moment. After 18 years of being told, no, daddy's earrings are too grown up for you, sweetheart, she finally got the wearables. It was a big moment for both of us. I also want to thank the funding sources. It takes money to do science. And in particular, this is also my disclosure. I have been funded by the chemical manufacturer, Novartis, Syngenta, but they since don't really hang out with me anymore. Somebody once said, if you're not pissing somebody off, you're not doing anything important. Based on the reaction I've got from the chemical companies, I'm a pretty important guy. So you guys are lucky to have me here tonight. I want to thank all of the students that have been involved over the last 20-some years, and everybody in blue here is an undergraduate. I had a wonderful experience as an undergraduate myself, and I've been doing my best to try to pay that back in my professional career. This is some of the recent students that have been involved directly in the research that I'll tell you about, and this is a photograph, the most recent I have. There is one guy, but he didn't show up for the photograph that day. And one of the other things that's incredibly important to me is to be able to work with a diversity of students, both first generation, low income, underrepresented groups, and in particular, several of the students that I work with, including 3 that are shown here, grew up in families that work in agriculture, that were migrant farm working families, and that's given me a completely different perspective on the kind of work that I do. Finally, I want to dedicate this to my grandmother, who passed away 12 years ago, and I was lucky enough that before she passed away, she passed on to me her love of education and her desire to make the world a better place through education, and I'm forever lucky for that. Feel lucky for that. I also feel lucky because she taught me something else that you can't learn at fancy places like Harvard and Berkeley and the NIH. She taught me if you want to get a point across, don't lecture, don't preach, don't teach. Don't give a talk. Don't present. Just tell a good story. So I'm just here today to tell you a story. And my story will start and end with a little boy who likes frogs, because that's who you've invited to talk to you today. Little boy who likes frogs. My mom sent me this book when my son was born, 24 years ago next week. And she wrote a note in there. She said, "Dear Tyler," my son, she said, "this was your dad's favorite book." I don't remember the book. But my mama don't lie. If she says this is my favorite book, this is my favorite book. But what I do remember is that I've been trying to answer this question, what is a frog, as long as I can remember. As long as I can remember, I've been excited about these guys. And one of the things you may not know, one of the things that excited me as a kid is that they're incredibly altruistic. You see the one on the top there has hurt its leg, and the one on the bottom is nice enough to give it a ride home. You probably didn't. I have students that to this day still believe that that's what's going on there. Hopefully you're getting a better education here at Claremont. And the punchline to the joke, which I won't tell because it's probably inappropriate, is that see what happens when you help a friend, because they lay these eggs on the way home. and this particular species, the females all lay their eggs in these huge communal clumps. So there's probably 300 females have laid each of their little clumps of 2,000 eggs in this one spot in this pond. And what I became interested in is how this behavioral ecology, if you will, interacts with sort of environmental developmental endocrinology. Really integrative. So for example, the eggs in the middle can be as much as 10 degrees warmer than the eggs on the edge in this situation. That means that if a female, as a matter of her behavior and as a matter of the ecology, if a female gets there first, her eggs are going to be warmer than the eggs on the edge. That means that her eggs are going to develop faster. That means that her larvae, her offspring, are going to grow faster and potentially bigger. But it also means, at the time I thought, that she might be able to manipulate the sex ratio of her offspring, because in some fish and some reptiles, temperature determines whether or not you're male or female. So I got interested in, and I should have told you, you're going to see a lot of gonads tonight. You're probably going to see more frog gonads tonight than you'll see in your whole life. I hope it's a special night for all of you. So here's males, these are testes, these large ones are ovaries, and I got interested in if you raise the species in different temperatures, will you see different numbers of male and females? But I was also interested in the interaction between genes and the environment. So if they have sex chromosomes, what are the limits that they put on what the environment can do in terms of whether or not you become a male or female? So I looked for sex chromosomes. So here's what that looks like. It turns out frogs don't have them. And I want to catch you up on where we're at in my story. It's going to be chronological. See, we started with a little boy who likes frogs in my grandmom's backyard. And where we're at now is work that I did for my senior thesis at Harvard. That's why there's a lot of undergrads here, right? So you guys use Microsoft Word and Adobe Photoshop, and you know that function, cut and paste? This is where it came from, cut and paste. See, when I was in college, you had to cut and paste the thing on your page. This is a page from my senior thesis in 1989. There I am at age 19. There's my professor, Bruce Waldman. And there's Laura, who probably went on to do something else because she doesn't quite look as excited as I was to be in a swamp at 6 a.m. on a Sunday morning, but it was like a dream come true for a little boy who likes frogs, and really the start of my professional career. One of the other things that I dreamed about as a little kid was going to Africa. I remember my dad would bring home these National Geographic magazines, and I would fold out the maps and dream of going to this magical, magical place. And it was truly a dream. My father made $9,000 a year for a family of 5. The first time I ever got on an airplane was when I went to college. But as a graduate student, now that's where we're at, that's where this little boy is in the story now. As a graduate student, I not only got to go to Africa, I got to grow this weird beard, I don't know what's going on with that, but National Geographic paid for it. I got to be in the magazine, I got to be in the show, I got to be in a Toyota commercial. You can look it up, it's called Remarkable Men Drive Remarkable Toyotas. Google it, you can look it up. But it truly was a dream come true. In the literalist sense, I got to become that guy that I watched on TV. I got to go to Africa. And I worked in a place called the Arabuko-Sokoke on the coast in Kenya. And one of the cool things about working in Africa is you get to say things like Arabuko-Sokoke. How many people have ever said Arabuko-Sokoke? See, because you've never been to Africa. And while working in Arabuko-Sokoke, I discovered this frog, which is the kind of thing that if you're a little boy who likes frogs, this is like, you're like, wow, you just don't know what to do with yourself. The males and females look completely different. And so I brought these animals back to UC Berkeley, and by now I'm an assistant professor. That's where we are in the story. And I brought these animals just because I'm a little boy who likes frogs. I like, But as a scientist, I had to formulate a hypothesis. So if you're a scientist, you have to have a hypothesis. So here's my hypothesis about how they got to be different colors. We showed, by the way, now we had Adobe Photoshop, so cut and paste looks a lot nicer now, see? So this is the same frog photographed once a day for 6 days, and we showed that they all start out green. This was a new discovery. Nobody knew this. They all start out green, and the females change color. So our hypothesis then was that the hormone estrogen, and we don't know why they change color, but in terms of the ecology and evolution of why they change color, but we wanted to understand the mechanism. And our hypothesis then was that estrogen is probably involved. So the same way as a human, when you reach puberty, if you were a female, estrogen would make your breast grow. Our hypothesis was when these frogs reach puberty, they change color. And so we tested that, real simple. We have some fancy tools I'll tell you about. We also do a lot of simple stuff. If you're imagining how would you test that, we did exactly what you think. We just dipped them in hormone. So if you dip them in testosterone, nothing happens. And if you dip them in estrogen, they change color. Supported our hypothesis. Now here's where the story gets weird. You might be thinking it's already a little weird. Because on February 15th, 24 years ago, February 15th, 1993, and I will never forget the date because it's the day before this guy was born. I was at UC Davis. His head, it rounded up. He came out to be a good-looking guy. In fact, he's single, if anybody out there is. Yeah. I'm giving this talk at UC Davis about my color-changing frog. My wife's in the audience. She goes into labor. It's 100% true story. You can't make this stuff up. She goes into labor. I have to leave in the middle of my talk to drive down, February 15th. He was born the next day. I'm driving down, and between contractions, my wife says, "You should patent that frog." And I thought it was like some crazy hormone stuff. I said, "You can't patent a frog." And she called her brother-in-law, or her brother, my brother-in-law. And he said, "Oh, yeah, you can patent a frog. Heretofore to be referred to." He's a lawyer, so he was talking like that. And we called it the Hyperolus argus. That's the species endocrine screen, or the, he says. And this is where things get weird. But first, let me tell you why you patent a frog. So her idea wasn't so crazy, and it really was hers. That here's a control. They're all green. If we give them estradiol, estradiol is the natural estrogen in every vertebrate animal. It doesn't matter if you're a fish, a frog, a dog, a cat, a hog, or a human. If you're a reproductively mature female, this hormone circulates in your body, and it makes my frog change color. If you give them ethinyl estradiol, this is synthetic estrogen that's used in birth control pill, makes my frog change color. If you give them DES, a pharmaceutical estrogen that used to be given to pregnant women, makes my frog change color. If you give them DDT, which we were talking about at the table, an insecticide that actually mimics estrogen, my frog will change color. So it turns out that we screened dozens of compounds, and every estrogen that was known, or every estrogen that made my frog change color, was also known to promote breast cancer. So we had a little frog from the Arabuko sucoque that we could raise by the tens of thousands and just dip them in solutions, and we could figure, you could send me a sample of your water, I could dip my frog in it, if it changed color, I'd be like, you might want to get something else to drink. What's more is we showed that we could give these frogs tamoxifen and stop them from changing color, and tamoxifen was the estrogen blocker used to stop breast cancer at this time. So that's why we patented this frog. But that's also why how things got weird. Because that's when a little boy who likes frog, because I patented a frog because I thought that'd be cool, I got a frog. But then a little boy who likes frogs gets introduced to some grown-up words. And somehow, at least in my mind, grown-up words always come in two. I don't know, maybe they're scared to walk alone. In this case, the grown-up word was intellectual property. You see, if you think of anything right now, it belongs to Claremont College. If I think of something, it belongs to UC Berkeley. They don't help clean the frog tanks, but if I think of something, it's their intellectual property. So the university said, little boy who likes frogs, welcome to the real world. That's a good idea, but if you don't find out how to make money on it, we're going to sell it. So I had to make money on it. So my wife, who had an MBA and an MPH at the time, we sold it. And what we did is we sold a contract to this little company. I'm being obnoxious. The biggest chemical company in the world, Novartis, said, we want you to tell us if atrazine does anything to frogs in terms of endocrine, in terms of hormone action. And here's atrazine. And you know, I was talking to Priya, I think, about I hated chemistry when I was in college. You won't believe how much chemistry I do now. In fact, I hated chemistry much in college. I hated it. It was so nice I had to take it twice. And you know, I didn't fail. At Harvard, nobody gets an F. They give A, B, C, D, E. I spent a whole semester trying to convince my parents that E was for excellent. The point is, chemistry is not the end of your science career. You can go on and get a PhD in biology and, you know. At any rate, this is what it looks like. If you're chemically inclined, it's an herbicide, it's a weed killer. It's been used since 1958. We use 80 million pounds a year. At the time, it was the #1 selling agrochemical in the world. It's used in more than 80 countries, but now outlawed in all of Europe. Outlawed in all of Europe. Outlawed in all of Europe. That's a lie. That slide is not true, and I'm telling you that I know it's a lie because the company's lawyer writes me letters all the time. He's not here today. He says, it has not been outlawed in all of Europe, his letter says. It has been denied regulatory approval by the European Union. Right? Me too. I don't know what the difference is, but I know that this one pisses them off. So this is a slide that I use. Because that's the kind of brother I am. So they asked us to look at another African frog, the African clawed frog, Xenopus. You guys ever heard of Xenopus? The African clawed. You have some here. I'll tell you why. The African clawed frog is used in most developmental biology, but for an odd reason. Here's the reason. In 1920, you didn't know you were going to get a history lesson too. In 1920, somebody discovered that the human pregnancy hormone, HCG, will make this frog lay eggs. By 1940, this is a 100% true story, this frog was the pregnancy test. If you thought you were pregnant, you would go to the hospital, and they would take some of your urine, and they would inject it into one of these frogs, and if the frog laid eggs, well, you're either happy or sad, depending on your situation. And I tell you the story for three. I would never show you a slide unless I have 3 things to tell you, because otherwise I'm wasting your time. So here's the 3 things I have to tell you. I show you the slide for 3 reasons. One is it shows you the value of curiosity-based research, right? Who was the first guy who thought, hey, you know, I wonder what will happen if I inject pee into a frog? Like, how do you come up with that? That there could not have been any hypothesis involved in that discovery, but you see how important the curiosity-based research was. The second reason I tell you this story is it's another illustration of how similar our hormones are to frog hormones. The estrogens that make my frog change color are the same as the estrogens that promote breast cancer, and the hormone of pregnancy is so similar to this frog's reproductive hormones that it'll make it lay eggs. That tells you that when I tell you what atrazine does to this frog's hormones, if you don't care about frogs, unlike the young lady who introduced me, if you don't care about frogs, you should be thinking, what might atrazine do to me? That's why I call this a tale of toads and men. The frogs are telling us something about ourselves. I guess the third reason for telling you this story is when they figured out new pregnancy tests, people just threw these frogs out. So every other weekend I go to San Diego. There's African clawed frogs in San Diego, and I can collect them for free without having to pay some company for them, which I guess technically makes mine African-American clawed frogs. But that's a point that's not relevant to the story completely. Just another contribution that we make. Atrazine inhibited growth of the voice box in males. This is the first thing we discovered, is that if you put atrazine in the water, the voice box of the larynx doesn't grow. And this is bad news for the company, of course, because male frogs sing and females don't for the same reason that men have deeper voices than women. Testosterone. So these data implied that there was something wrong with the testosterone in these exposed frogs. And of course, there's something wrong with testosterone. Of course, you look at the gonads. I told you, you're going to see a lot of gonads today. And so here's Xenopus gonads. So these are the kidneys. They're internal, the kidneys, as well as the gonads. Here's testis. So it's a male, but oh wait, it's got company. It's got ovaries, then it's got another testis, then it's got more ovaries. This frog could hurt his leg and give itself a ride home, which is just not normal. And I say that because the chef was pointing out earlier today that there are fish that are naturally hermaphroditic. That's true. There are no frogs that are naturally hermaphroditic. And lots of people think that, and I didn't know why for like 20 years. Who knows why people think that? Somebody always knows the answer. Say it. No? Jurassic Park. So in the book and the movie, supposedly the dinosaurs changed sex because frog DNA made them change sex because frogs are hermaphroditic. That's what the book says. That's science fiction. We're going to stick to science today. So we had a hypothesis, because a little boy who likes frogs is a scientist. Now you got to have a hypothesis. And my hypothesis was if these males were growing female parts and not growing their male parts, then maybe something's wrong in the testis. So imagine that this big circle is not your testis, but somebody that you don't like, because we're going to subject it to atrazine. If you have a testis, you should make testosterone. Who knows what that word means? Like literally, who knows what the word testosterone means? I'm sure you've heard it. Do you know what it means? Who knows what a portmanteau is? Give it time. I didn't learn this word until I was 48 years old. You know what it is.
It's essentially a combination of two hormones.
Like smoke and fog, you get smog. Twist and jerk, you get twerk. Testosterone is a portmanteau. It means testicular hormone. So it is literally the male hormone. So our proposal was that atrazine turns on aromatase. Aromatase is an enzyme. I want you to remember, it's going to come up again. It's the machinery that converts testosterone into estrogen, another portmanteau. Estrogen means the generator of estrus. So it's the female hormone. Now, this is fine if you're a female. If you're a male, this means that you've been demasculinized because you're using up your testosterone, and you've also been feminized because now you're making estrogen. That was our hypothesis. And so we tested this. We measured blood levels of testosterone, and these are daytime levels. They get much higher than this. But these are control males, so unexposed, atrazine-treated males, and they're no different from a female. By now, here's where we're at in my story. Company's upset. They don't want to have anything to do with Tyrone anymore. I published it anyway. And I published it, so here it is. Hermaphroditic demasculinized frogs after exposure to the herbicide atrazine at low ecologically relevant doses. That's probably one of the most offensive titles I could give to the company, but that's the kind of brother I am. And not only did I published in Proceedings to the National Academy of Sciences, PNAS. You guys know PNAS? It's a big deal. And now, here's where we're at in the story. A little boy who likes frogs about to come up for tenure. And I published a paper. PNAS, one of the top 3 scientific journals in the country. So I call my mom. I call my mom every Sunday. I said, Mom, I have a paper coming out in PNAS. Silence. I said, Mom. She said, no, I heard you. I just don't understand how you get a paper cut on your penis. I said, no. I said, penis. And it's a 100% true story. The next Sunday, my mom calls me back, and she says, how important was that paper you published? I said, really important. And she said, because I went to Barnes & Noble. They never heard of that magazine. This is now my most important publication. It's a kid's book that I didn't write, but my mom could get it in Barnes & Noble. And at the end of the talk, I'll tell you why that's so important and how that's changed my view of the responsibilities of a little boy who likes frogs. That'll be the end of the story. By the way, 4 black men and a Latina published this paper. That's another important thing. That's probably a record for the National Academy. That's an important thing for me. As important as it was, we didn't know if these hermaphrodites were males with ovaries or females with testes. Because I discovered as an undergraduate that frogs don't have visible sex chromosomes. But we had a good idea because if we expose these frogs to atrazine, we might get 50% females, 30% males, 20% hermaphrodites. So we had a good idea it was the males, and that fit with our hypothesis of how atrazine works. But we also didn't know what happens when these animals become adults. And that sounds also like an easy question to answer, except that at the time it took us like 4 to 5 years to grow these animals up. That means you gotta get an undergraduate as a freshman and say, hey, I have a good idea for you. And maybe by the time you graduate, in fact, it was worse than that. It took us 8 years. And here's what we discovered. We grew them up, and they hurt their legs all the time, you know. And by the time the paper came out, a gene called DMW, so this is PCR, so we did some fancy stuff. So we could look at the genes and tell, even though they don't have sex chromosomes, whether or not an animal's a female. If they have 2 bands, they're a female. If they have 1 line or 1 band here, then they're a male. So for example, the guy with the hurt leg who looks like he's smiling, he's a male, and that's his brother. So it turns out, in fact, that like 10% of the exposed animals completely turn into females. There he is laying eggs. He's a grandmother now. There's something you don't hear at Thanksgiving every day. He's a grandmother now, completely indistinguishable from a genetic female, but he doesn't have the female gene. Atrazine has completely converted it into a female. Now, I probably could have published that, males turning into females, but I had tenure. I was in no hurry. I wanted to know what happens to the other 90% of the males that don't turn into females. Are they competitive? Can they breed like a male? The problem is these frogs breed in the spring, and apparently so do undergraduates. They all want to go away to spring break to some pool party thing or something like that. So I got to figure out a way, because I'm working with undergraduates, to get them to stay around spring break so we can figure out what these frogs do during the spring. So I said, how about in 2008, this is how far back we're going, how about I have the pool party? Snoop Dogg won't be there, but you can have the pool party and you'll get a PNAS paper. How many people would do that? How many? None of you? You'd do that, right? Okay, went something like this. This is our apparatus. And the idea was, and I just made this up. I said, look, how about if we put 4 control females in here, 4 control males, and 4 atrazine-treated males? And I noticed like the sex ratio guys that you want at the club, but the idea is the females are supposed to be limited to see if these atrazine-treated males can compete. I just made it up. So we did this 5 times, but one of the times one of the students, kicked the tank, so I couldn't do it. So what you do is we put them in, so there they are in the pool. So you put them in at 7 p.m., and then there they are, they're swimming around, and then the lights go out. You put on a little Marvin Gaye. You young people don't know nothing about that. And the next morning, you come back, and see, we stitched them so we could tell who's who. And you just look at who got the hookup and who didn't. It's real simple. And when you do that, you find out that, and again, we did it 5 times, but one morning one of the students kicked the pool, so we only have 4 nights of data. And we couldn't retest people. We wanted everybody to be a virgin and all that stuff. So it turns out that only 2 males, atrazine-treated males, ever got the female. They couldn't compete. But now I'm an endocrinologist, so I can't just observe. I got to measure something. I got to measure a hormone. So imagine you're at the club, the lights come on, somebody's going around writing down names of who's with who, and then they pull you apart and stick a needle in your heart. That's how this ended, because that's where we get the blood from. And so it turns out, as you might guess, the controls, the unexposed animals, on average have much more testosterone than the atrazine-treated males. But what's more important is if you look at the individual levels, if you look at who made the love connection, it's like these guys don't have enough testosterone to appeal to the females, or they're getting beat up by the other males. We don't know. And we also didn't know at this time that the guys with the female have high testosterone levels because they got the female, or did they get the female because they had high testosterone levels? Turns out now we know they got the female because they have high testosterone levels. Whether or not you get the female depends on your testosterone levels the night before. And these atrazine-treated males have such suppressed, low testosterone levels that they're just not competitive. There's almost like a threshold cutoff. They just can't do it. So that was pretty interesting. I could have published that. But again, I had tenure. I was in no hurry. I wanted the whole story. I wanted to know they're not competitive. Are they competent at all? So then I did what I called the Motel 6 experiments. In this case, I just got them a room, and I just said, okay, here's a female with control. Here's a female with an atrazine treated. Let them have a night. Collected their eggs. And then we just asked how many of these eggs were fertilized. So it's kind of a measure of sperm count. It's a measure of fertility. So this egg is infertile, and these are examples of ones that actually went on to develop. And it's a real complicated procedure to figure out the fertility rate. I'm being obnoxious. There's an undergrad sitting in the room going, 1, 2, 3. I think he majored in math. So if you do that, though, you get a lot of data. Controls fertilize about 85% of a female's eggs. Atrazine-treated males only like 15%. And you know why? 2 reasons. One is they don't even try. There's no competition. They just sit there and watch the females lay eggs. They're not even interested because they don't have enough testosterone to show male behavior. The other one is if you open them up and slice them up and look under the microscope, their testis looks like this. How many people do statistics? No statistics. You like p-values? I love p-values. But you know what I love even more is when you can look at something and see that it's different. And we measured it and did some MANOVA and looked at the residuals and all that stuff that people do. But you could see the difference. I literally gave photographs to students who weren't involved at all in the project, because this went on for years, and said, put these into 2 piles, and you could do that. Because what you're looking at is a slice through a testis and a control, and you see this testicular tubule, you see all that stuff, and there's soldiers ready to go. That's sperm in there. If you look at the atrazine-treated males, right, there's the outline of testicular tubule. They just have cellular debris. Not only do they not have enough testosterone to show the behavior, but even if they did, they don't have enough testosterone to maintain sperm production. We measured a whole bunch of other stuff, and we published that. We called it Atrazine Induces Complete Feminization and Chemical Castration in Male African Clawed Frog. Chemical castration. Chemical castration. It's a grown-up word because it's in 2. Chemical castration. The company hates the word chemical castration. That's why I put it in the title. That's just kind of brother I am. The other thing that's important, The other thing that's important here is that there's 1, 2, 3, 4, 5, 6, 7 undergraduates co-authored this paper. Every one of them has a PhD or an MD right now. I'm proud of that. Second PNAS paper. The next thing that happened was, I got tenure, by the way. The next thing that happened was, otherwise the company would have seen to it that I'm not around in more ways than one. The next question we asked is, do these effects occur in other species of frog or just in the African clawed frog? And so we looked at a North American frog, and the North American leopard frog. And here's the testis. I told you, you're going to see a lot of gonads tonight, but you won't see a lot like this outside of my lab. See all this? I call it junk in the trunk. Those are eggs that are yolked up and bursting through the surface of this male's testis. So it's not a hermaphrodite. It has testes, this other species, but it's, instead of sperm, it's making eggs, and they're yolking up, called vitellogenesis, bursting through the surface. Now, at this point, the Environmental Protection Agency becomes involved, because I write them an email, and I said, look what atrazine does, because I couldn't write to them when I was working for the company. I was under confidentiality contract. Confidentiality, you see, they didn't want people to know. So I wrote a letter, I said, it's an email, I said, here's what atrazine does. And I always say curfew, but that's not what I mean. I broke embargo, because this was about to be published in Nature. That's another one of those magazines that you can't get in Barnes & Noble, but it's a big one. It's like the #1, right? And the EPA wrote back and they said, well, it's interesting, Dr. Hayes. However, we do not see this as an adverse effect that would trigger reassessment and regulation of the herbicide atrazine. Not an adverse effect. Not an adverse effect. Not an adverse effect. My wife tells me, and she's going to kill me when she knows I show this photograph. My wife tells me that there is nothing more painful in life than childbirth. And I'm going to have to give her that because I'm never going to experience that, childbirth. But are the rest of the men in the room with me when I say that a dozen chicken eggs in my testicle would have to be in the top 5? Right? That's what I thought. EPA says, no, it's not an adverse effect. So we're still using it. Not an adverse effect. The next question we asked, though, is do these effects occur in the wild? Because maybe this is a laboratory artifact, right? We're doing this all in the lab, so maybe this is just a laboratory artifact. And so we went into the field, and now these are more gonads. And so now these are testes, these are kidneys. And if you slice this up, and if you're not used to looking at what we call histology, imagine I'm slicing up a salami and folding out a slice. So I'm going to fold out a slice for you. The color's different because of the stain we use under the microscope. I'm going to blow this up, and I'm going to blow this section up. And you should be familiar now. These are testicular tubules, just like we have guys. Except instead of sperm, you see, those are eggs. I published this in Nature. I call them testicular oocytes. You know, it's a grown-up word. Comes in 2. Testicular because you can't just say eggs and testes. You have to have a fancy word. Testicular oocytes. Testicular oocytes. The company, the lawyer, wrote a letter to my university. They wanted me to have my paper retracted. They said, because I made up a word. That's how desperate they were. They said, I made up a word. Aren't all words made up before they're a word? Plus, I went to Harvard. I can make up a word if I want to. So the idea was, so here's where most of the atrazine is used in red down to the gray-white. And we did this study where we control for latitude, if you're into the geography. And I'm just being obnoxious, actually. This is Highway I-80, and we were going to a conference in Indiana, and we collected frogs and water along the way. and got a Nature paper. See, that's fuel efficiency right there. And I was coming up for full professor, by the way, so this was important. And the point is, everywhere we found atrazine, we found feminized frogs and vice versa, but it was published in such a high-profile journal because we also had the lab data to back it up. It wasn't just this correlational analysis. We had lab data to show that this wasn't a normal aspect of development. And so in the last few minutes, here's where we sort of make the switch to A Tale of Toads and Men. This is Lake Nabugabo in Uganda, and you're looking at I believe, arrowroot crop where the runoff is collected in these containers and goes to the village for drinking and cooking and bathing water. And I show this because it illustrates not the similarity, but the oneness between environmental health and public health. If I told the men in this village, you know, the fish and frogs in this pond have this, they would clearly see the connection. Whereas in my village, so that's where the A's or somebody play, I don't really care. If I live somewhere there, my water just comes from there. I don't have to take a yellow bucket over there and get it, but we make big assumptions that we have this Environmental Protection Agency that's protecting us from bad things in the water so that we're better off than Lake Nabugabo, Uganda, and I don't think that's really the case. I call this From Silent Spring to Silent Night because in much the same way that Rachel Carson taught that silent spring and the death of birds due to pesticides was a warning to humans, I believe that our silent night and the 70% of amphibians that are declining globally is also a warning to us. A group of animals that survived the mass extinction that took out the dinosaurs are now 70% are in decline. A colleague of mine wrote in Ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. Well, I've talked to you about more than just correlation. I've talked to you about control studies in multiple populations, species, genera, different phylogenetic families of frogs, but it turns out that this effect of atrazine happens in every vertebrate class that's been examined, fish, reptiles and birds, and mammals, and that there's potentially an impact on us. I know this because I wrote to everybody in the world who studies atrazine, and I said, hey, let's write a paper. And so we wrote this big paper, 22 of us from 12 different countries, all studying atrazine independently. And in this paper, I invented the word gonadotoxin. They also tried to have that retracted. Yeah. You know, I read somewhere, I think in Science, another one of those big fancy journals, that all of language, like all of humans, came from Africa. You know, so I'm just doing what my people's good at anyway, making up words. That's what we do. So here's what we found out. So there's my frog testis, sperm, give it atrazine, no sperm. This is a guy in Belgium with fish, sperm in the testis, give it atrazine, no sperm. This is a reptile from Argentina. I didn't even know these people. This is a caiman. It's like a big alligator. Sperm in the testis, give it atrazine, no sperm. This work was done in Croatia and Nigeria. Sperm in a testicular tubule of a rat, give it atrazine, no sperm. This work was done in Pakistan, in quail, in a bird. Sperm in the testis, give it atrazine, no sperm. So people all over the world were showing this same thing. So it wasn't just a frog issue. Is it because testosterone declines when you're exposed to atrazine, like we showed in our frogs? Well, it turns out if you look at salmon, this is a guy in England, testosterone declines. Here's my frogs, here's rats, which are mammals like us. So in every vertebrate class where testosterone levels had been examined, you're also seeing the same effect. You can't do experiments on humans, but sort of inadvertently, they're done. So this is a colleague of mine that studied sperm count in Columbia, Missouri, and here's control males. And these are men who have trouble getting their wives pregnant and have a low sperm count. They have significantly more atrazine in their urine. So if you have their levels 0.1 parts per billion, that's like 1/1000th of a grain of salt in 2 liters. And that's what we use to chemically castrate frogs and fish. So if you have enough atrazine in your urine to chemically castrate a frog or a fish, and you don't have sperm. And we know that atrazine causes a sperm decline in every animal that's been examined. What's more is, now watch, because I'm going to squash the graph down, so don't let me trick you. And I squashed it down, you can see the numbers are still there. Because here are atrazine levels in men in California who work in the fields. And I'm going to squash it down again. Because here are atrazine levels of men who apply atrazine in California. Men who apply atrazine have 2,400 parts per billion atrazine in their urine. That's 24,000 times what we know is associated with low sperm count in Columbia, Missouri. That's 24,000 times what we use in my laboratory to chemically castrate frogs and fish. One of these guys could pee in a bucket, and I could dilute the atrazine in their urine 24,000 times and use the atrazine in their urine to chemically castrate and feminize 24,000 buckets of 30 tadpoles each. So now a little boy who likes frogs gets introduced to something called environmental justice. Environmental racism. 2 words, so you know it's grown up. Because 90% of the workers in California are Latino. I'll come back to that. Does atrazine induce aromatase, that enzyme I told you about? In frogs, it causes egg yolking and egg production, which you don't have to worry about in humans. But in humans, aromatase is important in breast cancer and in prostate cancer. With regards to prostate cancer, there's an 8.4-fold increase in prostate cancer and men who work in their factories bagging atrazine. In a community their factories in, that's 80% black, 80% African American. What's more is there's at least one study that shows that there's a correlation between breast cancer and women whose well water is contaminated with atrazine. P less than 0.0001. I went to visit them. They changed the name to Syngenta, which I think they should spell with an I instead of a Y, but nobody listens to me. They have a pipe that flows into the Mississippi River. 1.2 million pounds of atrazine flow into the Gulf of Mexico every year, which I don't think anybody's studying it, in a community that's 80% African American. And I bring that up because it's another environmental justice issue. These are the top 13 cancers that you're going to get in America. In red now are the ones that you're more likely to get if you're African American, if you're black. What's more, if you compare mortality rates with Caucasian or white Americans, you're more likely to die from 13 out of 13 cancers. And this is corrected for socioeconomic status and access to health care. You're more likely to die from 13 out of 13 cancers if you're black or African American, and there's similar data available for Hispanics. And my colleagues who study cancer tell me that less than 30% of cancer is predicted by genetics. So that means that when the doctor tells you you're more likely to get breast cancer if your mother, your sister, your daughter, or somebody in your family has breast cancer, they're not telling you you have bad genes. They're telling you you've been exposed to the same crap that the rest of your family's been exposed to, because if you're a minority or low income, you're more likely to live in and more likely to work in areas where you're exposed to chemicals that we know are associated with adverse health outcomes. And I'm all about Komen for the Cure. They ask me to give talks every now and then. But with the exception of HeLa, none of the cell lines that we use to study cancer come from black or Hispanic men or women. So even if we find a cure, it may be irrelevant to the people who are more likely to get and more likely to die from the cancers that we're trying to cure. The problem is there's no money in prevention. You can't put your name on prevention. You can on a cure. We showed, as well as another lab, that if you take human cells and expose them to atrazine, they start expressing aromatase and making estrogen. Just like we've shown in fish, just like we've shown in amphibians, just like we've shown in reptiles, just like we've shown in birds, just like we've shown in rats. Surprise, surprise, human cells do the same thing. In fact, one of my students showed that if you give a breast cancer cell atrazine, it starts making aromatase and producing estrogen. Now, that's serious because breast cancer is estrogen dependent. Even though most women get breast cancer after menopause, when the estrogen levels in your blood are lower than they've been in your life, the likelihood of breast cancer depends on lifetime exposure to estrogen and also local expression of aromatase. So the damaged cells make their own estrogen, even though it's low in the blood, and that causes these cells to turn into tumors and spread. In fact, the #1 treatment for breast cancer right now is a chemical called letrozole. And letrozole works, this drug, by knocking out aromatase and decreasing your estrogen so that your cancer doesn't grow. How much sense does that make when the #1 contaminant of drinking water does the opposite? Turns on aromatase, increases estrogen, and increases the risk of breast cancer. It made sense to somebody. Because see, Novartis Oncology offers treatments that range from breast cancer, et cetera. So the same company in 2000 that gave us 80 million pounds of atrazine, which turns on aromatase, also sold us a drug that blocks aromatase to treat breast cancer. So if you were living in the Midwest taking letrozole to treat your breast cancer, it was having to fight it out with a chemical made by the same company. I published a paper called The One-Stop Shop, Chemical Causes and Cures for Breast Cancer. And the company got upset. I don't have to tell you the rest of that story, how that ended. So I think what's happened is my interest in this aquatic organism, little boy who likes frogs, has told me a lot about this aquatic organism. We grew up in an amniotic fluid. We depend on testosterone, estrogen, thyroid hormone, cortisol, exact same hormones that my frogs depend on. And we now know that you and your children will be exposed to over 300 synthetic chemicals before you leave the womb. And most of them, we have no idea what they do. The best thing about atrazine is that we know. We know that atrazine in rats causes prostate and mammary cancers. It's associated with it in humans. We know that atrazine in rats causes immune failure. It's been published. We've shown the same thing in frogs. We know that atrazine causes neural damage when rats have been exposed in utero. And we know 3 things now that I'm going to show you that moved me in a very special way, that showed me that I have to be much more than just a little boy who likes frogs. An EPA laboratory showed that if you give rats atrazine, they're more likely to have an abortion because of the hormone imbalance it creates. A second EPA laboratory showed that if those rats that don't abort, the sons are born with prostate disease. The sons are born with the prostate of an old man. Another EPA laboratory showed that if those rats that don't abort, the daughters are born with poor mammary development, poor breast development, such that when they grow up, their offspring have retarded growth and development because they can't be fed properly. And more than anything I did in my frogs, this study moved me. Because see, the rat on the bottom never saw atrazine. Rat on the bottom was affected by atrazine that its grandmother was exposed to. The rat on the bottom never saw atrazine. was never exposed. The rat on the bottom was affected by atrazine that its grandmother was exposed to. The rat on the bottom never saw atrazine. The rat on the bottom was affected by atrazine that its grandmother was exposed to. So when I think about my little girl, who's 21 now, when I think about the fact that my grandchildren could be affected by chemicals that my daughter was exposed to in the womb, your grandchildren could be affected by chemicals that we're using today. My grandchildren could be affected by chemicals that we're using today. And that moved me in a very different way than just a little boy who likes frogs. A colleague of mine showed that if you get pregnant during peak atrazine contamination, you're more likely to have a baby with birth defects. I won't show you all of them, but I'll show you one that's important to me, and I apologize for the graphic images. So this is a case-control study of maternal residential atrazine exposure in male genital malformations. This wasn't work that I did, but this study showed that if you get pregnant with a son and you're exposed to atrazine, you're more likely to have a baby with hypospadias. That's what a urethra doesn't end through the penis. You're more likely to have a baby with cryptorchidism. That's what the testicles don't descend into the scrotum. You're more likely to have a baby with micropenis. That's what a phallus, the penis, doesn't grow. And I say this is more interesting to me because male genital development in humans depends on testosterone. And if you're exposed to a chemical that limits testosterone in every animal that's been studied, you look like you have a baby that didn't get testosterone. And we know that excess estrogens like DES can cause this malformation. And if you're exposed to atrazine, which induces estrogen, you look like you have a son that's been exposed to too much estrogen. So in terms of environmental justice, I think we have to stop using I use this guy because he works for the company, doesn't like me, but we have to stop using the adult white male to determine acute toxicity. It just doesn't cut it because we know that something that doesn't hurt an adult white male can cross the placenta and do harm to a developing fetus that's counting on delicate balance of hormones. We know that even after the baby's born, chemicals like atrazine can come across in the milk and affect these developing infants that are still depending on hormones for their development. And we know that a little bit of toxin to an adult is a lot of toxin to a little one. And we have no real test and validation set up now to evaluate chemicals in this way. The EPA said about my work, because they kept it on the market for 20 years since I discovered these things. They didn't say it didn't do anything. They said, quote, a monetary value is assigned to disease, impairments, and shortened lives and weighed against the benefits of keeping a chemical in use. A monetary value is assigned to disease and impairments in a country where we assume that everybody's created equal, but certainly afterwards, they're not treated equal. As a matter of environmental justice, I'll tell you, you may know because you live in California, but we'd be the 5th largest economy in the world. That's why we can talk about CalExit. If we were our own country, we'd be the 5th richest country in the world. Not because of Hollywood, not because of tech, not because of oil, because of agriculture. 1 in 10 jobs are in agriculture. 30% of the land's in agriculture. 350 agricultural products come out of California, and we produce half. How many people knew this? 50% of the U.S.'s food comes from California. 50% of everything the U.S. eats comes from California. As a result, we use more pesticides than any other state, and 90% of the workers are Hispanic. And if I put in red here now the top 10 counties for agriculture, these are the 10 counties that make us the 5th richest country in the world. Where do you think the 30 poorest towns are in California? So the people who make us the 5th richest country in the world are the unintended targets of chemicals like atrazine, like chlorpicrin, which was originally developed as a nerve gas, like 2,4-D, which is a component of Agent Orange, used to kill food to starve people. The people who make us the 5th largest economy in the world are the unintended targets of chemicals that we know are associated with adverse health outcomes. And as far as the argument goes that we have to feed the world, atrazine is used on corn. Here's where the corn goes. What's missing from this pie chart? Food. We eat so little of the corn we grow, it doesn't even appear in the chart. We eat less than 2% of the corn that we grow. Atrazine increases corn by 1.2% in a world where 20% of the people die of starvation. Yet we accept this monetary cost, and we accept it because 90% of our seeds in blue are owned by 6 chemical companies. So they're in the process of producing GMO seed so that they can sell more chemical. That's problematic. It's also problematic that the head of the FDA is a former Monsanto vice president. That's a conflict of interest. So is owning a chemical company and selling seed. My professor told me when I started working on this, don't be an advocate. He said, let the science speak for itself. Somehow people think that if you're a scientist, you can't advocate. I was told I have to walk across this when I talk about crossing the line. I believe very differently now. I believe differently because here's what Syngenta says on their website. They assume no obligation to update forward-looking statements to reflect actual results. One is, who says shit like that? But two, more importantly, here's what the EPA said about my work in 2006. They didn't say that it was wrong. They said the ultimate decision is much bigger than science. They said the ultimate decision is much bigger than science. It weighs in public opinion. And I immediately thought about my mom. I'm getting tenure for publishing in fancy journals that my mom can't get access to, and even if she could, she couldn't read it. But the EPA is counting on my mom to have an opinion, to tell it what to do. So we have to step out of the ivory tower and make sure that our work and our ideas are published and available in much more important places than the places we reward each other for. Finally, the EPA has announced that atrazine is bad for the environment. It's a reproductive toxin. I told them that 20 years ago. And I've changed my philosophy now about being an advocate. You can't just let the science speak for itself. You have to be out there. I follow a couple different philosophies from great thinkers, and I'll share those two with you in conclusion. One is, someone once said, those who have the privilege to know have the duty to act. If you're sitting in this room right now, you're like me. I grew up low income, black community, but I've been to Harvard, I've been to Berkeley. I'm here with you now. If you have that privilege, you have a duty. This guy said that, by the way. And finally, I want to share one other great thinker who said, it's time for us as a people to start making some changes. Let's change the way we eat. Let's change the way we live. Let's change the way we treat each other. You see, the old way wasn't working, so it's on us to do what we got to do to survive. And more than ever right now, I think these are words that we need to listen to. This guy said that, by the way. Thank you for your time. I'll address questions, comments.
If you have a question, please raise your hand and Michael or I will come to you with the microphone.
Hi, thank you for speaking to us tonight. So I was just wondering if there's any sort of like preventative measures you could see, like taking the, like putting your sperm in a sperm bank, for example, or taking that like breast cancer drug preemptively. I just mean like, I'm just spitballing here, but.
I mean, there are many things you can do, but I would suggest the more important things you can do are to first off be an advocate for yourself and an advocate for others. One of the things that has, and no disrespect to anybody, that annoyed me the most was when I was, I will protect the county. When I was invited to a fairly affluent county to talk about an issue where they wanted to spray pheromones for the light brown apple moth or something like that. And my attitude was, if you were one county over protecting the people that grow our food and are at risk every single day, then nobody would talk about spraying over your house. So one of the things I think is to be an advocate for all of us and not just for ourselves. So somebody will always ask about a Brita filter. Brita filter will take atrazine out, but it doesn't protect the people that are most affected, because those high levels in factory workers and farm workers are from breathing it in and across the skin. So even if they had the information and even if they had access to the money to get filters, it still wouldn't hurt the people, help the people that are most effective. So being an advocate for yourself is most important. Speaking out, voting for things like GMO labeling. I have no feelings, I've seen no data that GMO is bad for your health. But 90% of, when I was a freshman, when I was some of your ages, the promise of GMO was higher nutrition corn and drought-resistant wheat and frost-resistant strawberries. But the technology has not been used for that. 90% of GMO products are to make the plants resistant to chemicals so that we can use more chemical. And so the issue is, I think, voting for things like labeling, buying locally, which especially here, you have the ability to buy local and to buy organic doesn't necessarily mean pesticide free, but it's voting with your dollars and saying that we want something different to happen in the world that we're in right now. Writing to the EPA. Every time they review a chemical, there's a public comment period. That's what that statement was about. I didn't know this. Writing to your congressperson. So those are the things that you can do, I think, that are preventative, things that will affect globally the environment and other individuals. And those are the things that I encourage you to do. And I never thought as a little boy who likes frogs that I would ever say things like, write your congresspersons.
Has there been any response over the years to the information that you've published for the agricultural area to make changes to drop this pesticide and use things that aren't so bad?
That's a long conversation. And no, it's in part because of that grid or that map that I showed you, and that's because 90% of this, if you're a farmer, 90% of the seeds that we use to grow are owned by 6 chemical companies. In many cases, you cannot buy the seed without, for example, fungicide. It comes already on the seed. And because you're buying from it, or for example, if you're buying Roundup Ready corn, you have to use glyphosate on it. It's designed. If you're buying 2,4-D resistant crops, you have to use the pesticide. And part of the problem is if I'm a farmer and you're a farmer, and I decide that I'm not going to use Roundup Ready corn and you're going to use Roundup Ready corn, I'm at risk for 2 reasons. One is, if your GMO corn blows onto my property, I can lose my property because I didn't buy the GMO corn. The other one is, when you spray your Roundup, I don't have the GMO resistant crop. Your drift from your crop is going to kill my crop. And so what's happened is 6 chemical companies, soon to be 4, because there are 2 mergers coming up, have captured the food industry in such a way that there's no way that you can pull out of it. And it's very clever if you think about it. But the conflict of interest is they not only want you relying on the seed, so you can't save seed, for example, you can also lose your farm, but they also want you to depend on the chemical that is what the company actually sells. Now you combine that with corn. The majority of corn is used for ethanol production. So when the conversation happens, it's not even about atrazine, it's about renewable fuels. And the first presidential caucus is in a corn state, Iowa. You're not going to go to Iowa and say, I'm going to appoint somebody to the EPA that's going to regulate atrazine, because now you're going up against the ethanol lobby, which that's a whole nother, I never thought I would talk about things like that, but that's a whole nother conversation, right? Not the least of which is corn is very efficient for ethanol production, and not the least of which is they produce the ethanol from the corn by burning coal. I mean, right? So there's some estimates that suggest that there's no net gain, that the amount of fossil fuel that you burn to produce the ethanol cancels out the ethanol, the energy in the ethanol.
Thank you very much for your talk. Impressive. Also in Europe, not everything is better. So I'm from Europe, but one point which is a super interesting story, and I think that matches the idea of the grassroots movement which is coming from the US. There's a small little town and community in South Tyrol, the German-speaking part of Italy, where they have a huge apple production. So pretty similar to Central Valley in some aspects of the agrochemical industry. What they did, they had an analysis on their community, on the playgrounds, in the schoolyards, in the church. They found agrochemicals. And then they had a vote. There was maybe 10 farmers and a small community with 1,500 people, but more than 80% of the population voted to have an entire ban on the community level, and it worked. And it worked. And it's the first place in Europe which is agrochemical free by a vote. Nobody knows how it will evolve. It will have some impact, but a lot of the farmers say we can grow up our apples without the agrochemicals. And that might be a way totally along what you're saying. I think it's not, if you see like Bayer and Monsanto coming together, it's basically the local people who have to change that. I don't see another way, especially if you see what you were saying that we have nowadays, where beyond facts we have, how is this, alternative facts, whatever. We don't have that in Europe now, but it might come.
Well, Europe in general is much more progressive. And I always say, maybe I shouldn't say to a European in the room, it's because you've had so much more time to screw up the environment, you're much more cautious now. But things like the precautionary principle, for example. So the reason that atrazine is banned there is in the US, a chemical has to be, you have to prove that it's bad, otherwise it stays on the market. Because most of these chemicals, there was no EPA when they came on the market. The EPA didn't exist in 1972. I showed you atrazine came on the market in 1958. So the EPA's challenge to screen and review 80,000 chemicals that had no regulation before they were used in the environment. The European Union has if the chemical's in the water supply, then you have to prove that it's safe or it's gone. And the fact is the company can't prove that atrazine's safe, so now it's gone. In the US, you're innocent until proven guilty. So the company's charged with proving that the chemical's bad. You see, there's a problem in how and how the system works in the US. Others?
I'm curious. So my impression is that a wide variety of pesticides are also endocrine disruptors.
Yep.
So I'm curious that if we were to successfully get rid of atrazine, do you think like that would resolve the problem? Is atrazine like worse than other chemicals, or is this just like the target that you happen to like have started with?
Again, the best thing about atrazine is that we know it's bad. And what makes it significant is it's widespread. It's the most common contaminant of drinking water, surface water, groundwater, rainwater, clouds. You find atrazine everywhere. It can travel 1,000 kilometers, that's 600 miles for Americans. So that's what makes atrazine the poster child, I think. The problem is with endocrine disruptors is it's not like, I don't know, it's not like I spray you with something and you die. And that becomes pretty obvious, like, oh, we need to review that. You're now, smoking is a perfect example. Look how many years, and we're still fighting about cigarettes. You don't suck down a cigarette and die, right? I mean, eventually, but not right away. And so the problem is, like with DES, that chemical I had up, it was given to pregnant women in my mom's generation, and then women developed uterine and vaginal cancers and ultimately breast cancers, but like 30, 40 years later. So now you had to go back and trace something back that they were exposed to in the womb, but at least there, the women had a prescription for it, and you knew. For something like atrazine, and it means you have to pick the chemicals one by one and do controlled experiments to figure out what to look for in the first place. Because otherwise, I mean, like if you get afflicted with whatever, you name your cancer, you may have been predisposed to a chemical that increased your likelihood. There's almost impossible to figure that out. And certainly almost impossible to prove it in a court of law. So that's the difficulty. There may be many, many other chemicals that are endocrine disruptors, and we know of some that are, but finding those and identifying those and showing in humans whether or not it causes a problem is very difficult. Atrazine's the best case because there's been so much done on atrazine, and still, for 20 years, it's still on the market, and it's still in the market. California's actually now listing it under I think Prop, is it 65? So it's now listed in California, but that's the most that's happened anywhere at any. Well, Wisconsin bans it county by county, but other than that, it's still there. Anything else?
Oh, hi.
Yes.
So you mentioned, obviously this is a very important idea, and you mentioned the necessity of being advocates.
Yes.
And so a lot of the problem with sort of putting ideas like this out in public consciousness is you publish this in PNAS, but scientific journals are not accessible to most people, and also from sort of a monetary standpoint, and also from just like a reading standpoint, hard to make it through. But the sort of the converse of that is when scientific articles get picked up by a news agency, there's a tendency to oversimplify and you lose accuracy. So sort of in the scientific world, and also I very much appreciated your presentation because it was very informative, but it was very entertaining. So thank you for that.
Thank you.
But sort of, I think you're a great person to ask. Have you found strategies for sort of fitting that gap of maybe extending your reach of where the research is, sort of the avenues and the places that people see it, while also maintaining accuracy and readability?
This is part of the problem with the ivory tower syndrome that I talked about, but because yes, I've taken many opportunities to try, and I'm not saying I do it well. Things like, the kid, I might throw it out and say it's a kid's book. It was an award-winning kid's book. It wasn't written by me, but the woman who wrote it spent 4 years with me, and that's a commitment. Like she went in the field with me, and she was in the lab with me, and it's taking away time from what the university says really counts. See, that doesn't really count. Giving a public talk doesn't really count. Being in a documentary film doesn't really count. And so that's part of the problem is that in the academic institution, the things that I would say count most, like what you're asking, things making your science available to the public, at least at Berkeley, don't really count. So documentary films are a number of ways to do it, and I've been in a number of films. There's a number of films now still being made because that tends to, more people will watch a film than they are pick up a paper, even a news article, for example. And also working very closely and getting training and knowing how to work with reporters so that thing doesn't happen. And sometimes it does happen. That probably what can be the most damaging and the most useful tool are things like the internet. We were talking about this at the table, that information can spread so fast over the internet, but also misinformation. And a number of things that I've been linked to on Facebook, I was like, no, I never said that. I mean, where did you get that from? And that, I think, is the bigger problem, is how do you help citizens differentiate between something that might be hype, something that might be false, something that might be misrepresented, and actually get to the real science. And I don't know that I have the answer to that yet. It's a goal of my career. Yes? There's no such thing. So it's a half-life of atrazine. Some papers will tell you 8 days, because if it's in water, under UV light, et cetera. In other places, like in France where it was banned, last time I saw a talk on it, hadn't changed in the aquifers for a decade. So when it's underground and not exposed to UV light, it can last who knows how long. I've personally seen a site in Tasmania where there was a plane crash carrying atrazine, and in an area probably twice the size of this room, nothing's grown there in years. So it really depends on, and I've seen somebody mentioned Nebraska, I've seen a field in Nebraska that got, here's a scary story, that got decommissioned and the farmer disappeared. And it's a field that we worked on and published on the condition of the frogs in that field. The last time I was there, which was probably 5 years after they had decommissioned the field, nothing was growing there. Because that's what atrazine does. It prevents weeds from coming up so that you can plant these big fields of corn. So I don't know. You get all kinds of answers from 8 years. I can tell you in France it hasn't changed in a decade, last I heard the story. Anything else? Oh, yes.
Yeah, I appreciate your talk, and I really like the term and terminology, chemical castration. And I think that maybe language can be very powerful in this battle, but it just seems overwhelming, the apathy that most individuals have. A lot of times they don't have the time to really digest this information, and they don't have, the practical language like the scientific journals. Most people can't really access that. So I'm just wondering, like you're kind of talking about advocates and like a grassroots movement.
Mm-hmm.
And I just wonder about the chemical companies. They need like an ombudsman or watchdog groups, which is, of course, they're not going to do that. The chemical companies have their spin doctors,
Yeah.
and they're going to confuse and refute that the evidence isn't all in, and it's going to be a continuum. And so I'm just wondering, how could we fast forward it to heighten awareness for the average individual? And what are some just no-nonsense things as individuals that we can do? I mean, like drink spring water. I mean, I know this sounds silly, but any suggestions?
I mentioned some some individual things earlier, like eating locally. I myself, up until the drought last year, probably grew 90% of my own food, and it was in many, many ways, a wonderful exercise of bonding and doing things with my wife and getting exercise and growing our own food, and also as a scientist figuring out what plants to plant where and when and all those kinds of things. So it can be a wonderful experience, and I always feel awkward saying that outside of California, because in California we can do that. We can grow things year round, here more so than even my place. So it's not a solution for everybody. Voting with your dollars, and i.e. deciding that you're going to buy organic and natural food. Europe does it. Europe goes beyond organic. They have biodynamic, for example, vineyards where they don't even water or use electricity, and they're still productive. So there are a number of things like that. And in terms of the industry, as some of you may know, I've experienced one of the biggest 20-year battles with them, both attacking me personally and threatening my family and trying to discredit me. And I make light of it here. But it's been a very difficult couple of decades, just being a scientist and being a father and a husband and trying to deal with somebody who's trying to destroy all the components of that, and they won't stop because it's about the dollars. In terms of the working with individuals, another thing I think that can be frustrating and confusing is there's so many things out there that are we worried about atrazine? Are we worried about Roundup? Are we worried about Monsanto? Are we worried about vaccinations? Are we worried about chemtrails? And sorting, are we worried about climate change? And again, also with the spin doctor saying, well, this one isn't real and this one isn't real. And also from the other side, I've seen people promoting things, like the vaccine thing, which was shown as fraudulent. But I still see these big arguments that vaccinations cause autism, and there's just no evidence to support that. So also trying to get at all of those things and trying to figure out how you sort through the information. And I think the spin doctors actually use that They say, oh yeah, we thought this was bad too, but look, this was wrong. So it can be very difficult, and I don't know that I know how to do it. I'm trying, that's my goal. More so than getting another PNAS paper, I assure you. Anything else?
Thank you so much for coming. I really appreciated your talk. I guess some of the ideas of the last few questions kind of have come together, and that quote from your advisor, "Let the science speak for itself." I mean, I think many of us in the scientific community have heard that a lot of times. On the other side, as you alluded to, you've been through this experience with the company, and honestly trying to work with them at the beginning, and seeing that as an opportunity to do good science, right?
Mm-hmm.
And I just would maybe like to hear a little bit more about that journey from, look, how can I do this science within the structure of working with this company, and then making the decision to step out of that and to do the science for its own terms, and kind of how that process went.
It was very abrupt in that, that's not true, I guess. So there was maybe once we made the initial discovery, there was maybe several months to a year where I tried to work with the company, where I said, we need to repeat the study, we need to increase sample sizes. And it became very clear that then the company didn't want things to happen quickly. And I now know that they knew that atrazine was problematic before they even hired me to do it. The strategy is you hire somebody who's young and up and coming, you get them hooked on the dollars, and they get to say to the EPA, we're spending millions of dollars to study this, and as long as you don't analyze the last sample, they can say it's in progress, and they're compliant. That literally is how it works. And so they have a number of people, I could call names, who have built their careers, and they don't really care if you do the work or not. I mean, look, I was fresh out of graduate school, brand new assistant professor. My wife had just started graduate school. They were paying me $1,000 a day. Literally, if I emailed and said, hey, I thought about atrazine today, I get a check for $1,000. So, and that's not to mention the money that was coming in my laboratory, and I never had students working on that project, just because I didn't want this confidentiality contract holding up their work. So it was all former students who came back to work for me. So they spent about a year, dragging their feet and not giving me money, and then I just finished it. I said, okay, we're done. And then they had a dilemma, because I said the report was done. So they had to try to find ways to say, well, the report hasn't been finalized. And so I quit. And after I quit, then they hired people to say, well, you did your statistics wrong, and they were analyzing everything to show why it wasn't done, and et cetera. And I just walked away. I thought, I could have stayed home and been a drug dealer or a prostitute. I don't need the money. So not that bad. And then at the time, shortly after that, they hired other people to do poorly done work to try to contradict my work. And then eventually after that, there was a long series of personal attacks that went on for 20 years, including threats to my wife and my daughter, and actually a series of over about 2 decades of things that, in all honesty here, let me see if I can pull this. So, and I pull this up because it's hard to talk about, because I think did we shut off? Oh, well, we're doing that tricky thing again. Because I think their strategy was to do things that were so crazy that if I talked about them, then I would look like I was crazy, right? And and I used to joke that they had this panel, because I won't repeat some of the things that they would say to me or write to me, but I used to joke that they had this panel on how to piss off a black guy, and they trained people on how to follow me around and do things, right? And so this is a quote from New Yorker magazine. So it turns out that they lost a $105 million lawsuit, but in that lawsuit, all of their private confidential documents became publicly available. And so stuff that I kind of knew all along, right, and I'll show you some of those, but the funniest part of the whole thing is, here's what they said to the New Yorker magazine. Their spokesperson, because the article was published in, it was a few years ago, but their spokesperson says, I am troubled by a suggestion that we have ever tried to discredit anyone. Our focus has always been on communicating the science and setting the record straight. Because I said they tried to discredit me, right? So here's documents now. She's troubled at the notion that they've ever tried to discredit anyone. So this is from their privileged and confidential private reading handwritten notes. Look at their strategy for science. The first thing they wrote down. So it's hilarious, right? So they actually sat in a room, and as part of their strategy, they wrote down, discredit Hayes. But what's more is they made this whole list of things, right? They had me psychoanalyzed. They said I'm paranoid, schizophrenic. They don't disrespect him, so at least they got part of this, right? And then they had this whole list of, pardon my expression, shit they were going to do to me, like buying my name on the internet, exploiting my father's health problems, investigating students, investigating funding, investigating wife, my family background, my school history. And one of my favorites is this one, set a trap. Somebody actually sat down in a room and wrote down that they were going to set a trap for me, right? So and they made their list of like risky, and they had their lists of risky and more risky things that they were going to do to me, and the people that they were going to pay to have those things done. And anyway. So, I mean, I think the whole idea, the plan, which makes me feel good as a scientist, that my science makes me feel that my science was so tight and so well done that they had to go after personal things and write down things like set a trap. I always tell people that, they went through all this Heinrich psychologist. The other thing they did was try to prove that I'm crazy. And I know this isn't nice and professional, but I always say I'd rather be crazy than stupid. And if you're sitting in a room writing something down like set a trap, right? I mean, because one, the first page of the Evil Spy Secret Handbook is you don't write it down. And if you do, you burn it and self-destruct it and eat it. You don't give it to a judge. If you're sitting in a room writing down something like set a trap, you're not only stupid, I'd say you're a special kind of stupid. but anyway, I won't go on. I laugh about it now, but it was a tough 20 years of also having things happen and not knowing. Like, am I paranoid or did this thing really happen to me? So anyway, I'll stop there.
On that note, that's all the time we have left for questions.
Thank you again.